Hello everyone, thank you for joining us for today's webinar titled A Genome-Scale CROP-seq Screen Reveals Mediators of T-Cell Signaling for Target Discovery. I'm Jackie and I'll be hosting today's session. We like our webinars to be interactive, so we encourage you to submit questions for our panelists at any point during the presentations using the Q&A box next to your slide window. We will address as many of these questions as possible during our Q&A session following the presentations. Please note that all attendees are on mute. This webinar is being recorded and we'll send you an email when the on-demand recording is available. A list of helpful resources and documents related to today's webinar is available in the resources list. And finally, to optimize your webinar experience, we recommend closing any unused browser tabs to maximize your bandwidth. And now I'm excited to introduce our speakers for today. Tilmann Buerckstuemmer is a biochemist by training. After a PhD at the Robert Koch Institute in Berlin and a postdoctoral stay at the Giulio Superdi Furga, he joined Haplogen as a principal scientist and later head of research. With the advent of CRISPR, Tilmann focused his team on CRISPR gene editing and quickly produced the largest collection of isogenic human knockout cell lines available to date. This became the basis for the commercial success of Haplogen Genomics and its acquisition by Horizon Discovery. At Horizon, Tilmann served as R&D Director and Head of Innovation with global oversight of all R&D projects. In early 2018, Tilmann decided to found Myllia Biotechnology, now Myllia Biotechnology, where he is currently serving as the CSO, together with his partners. He is passionate about science and enjoys working with multidisciplinary and multinational teams. Also presenting today is Angela Churchill. Angela is a product marketing manager for single-cell applications at 10x Genomics. In her academic career, Angela studied polycystic kidney disease using stem cell-derived kidney organoids as a postdoc at the University of Washington, and investigated the role of transcription factors during pancreas development as a graduate student at Columbia University. Prior to joining 10x, Angela was with Horizon Discovery, a Perkin-Elmer company, and Genuity Science in product management roles. So now I'll hand it off to Angela first to get started.
Hello everyone, my name is Angela Churchill. I'm a product marketing manager at 10x Genomics. It's a pleasure to be here to introduce this webinar with Myllia Biotechnology. Briefly, I'll provide some background on the 10x Chromium single-cell CRISPR solution and how single-cell CRISPR screens can be used to advance biology. As most of you are aware, CRISPR tools for engineering the genome are rewriting the rules of what's possible in biology across nearly every industry. From agriculture and energy to the next generation of therapies in medicine, there's great promise of progress. The programmability and ease of use of CRISPR to manipulate the genome is enabling new applications and insights not only for writing, but to truly unravel genetics' functional contribution towards disease and treatments. The flexibility and effectiveness of CRISPR at perturbing the genome has been applied across many research areas, contributing to wide ranges of discoveries in everything from optimizing the design of chemotherapy regimens to accelerating hit identification and lead optimization in drug development, and even to better understand how viral infection works across a variety of infectious diseases. As CRISPR has developed as a technology, different formats for applying CRISPR to study gene function have emerged, building a toolbox to investigate gene function. The traditional CRISPR toolkit includes three main categories. The first is single gene edit systems. These can allow researchers to model disease and are typically profiled in much greater depth. And then there are arrayed screens and pooled screens, where researchers can examine more genes, but the output is typically limited to changes in cell morphology or cell survival. These current tools, though impactful, impose difficult decisions between the scale of experiments and depth of readout, which leaves the toolbox incomplete to understand mechanisms driving the phenotypic changes for novel biological insights. Single-cell CRISPR screening offers the sweet spot of increased scale while also offering increased depth of readout that allows for deep understanding and characterization of gene perturbations at scale. Pooled single-cell CRISPR screens enable researchers to interrogate the function of hundreds to thousands of genes in parallel, while single-cell transcriptomic analysis provides a rich phenotypic readout of the gene expression perturbations being applied. Our 5' CRISPR kit is designed to be compatible with all the most commonly available guide RNA scaffold sequences with no guide customization required. That means if you've already constructed and tested a guide library for bulk or arrayed CRISPR screens, you'll have the flexibility of analyzing it in a single-cell format as is. Our single-cell CRISPR screening assay enables multi-omic readouts as well, allowing researchers to better understand the impact gene perturbation is having on the cell state. So in addition to whole transcriptome and guide RNA from the same cell, you can also obtain cell surface protein and immune receptor data. This assay has compatibility with CRISPR knockout, CRISPR interference, and CRISPR activation screens, providing the flexibility to investigate a wide range of gene functions. We see single-cell CRISPR screens as the next generation of the CRISPR toolkit, and it's already being utilized throughout disciplines to unravel gene function and understand contributions of key cellular processes in biology, from oncology to developmental biology. And now I'll hand it off to Tilmann Buerckstuemmer from Myllia Biotechnology, where we'll be hearing about their genome-scale single-cell CRISPR interference screen.
Hello, everyone. Good morning and good afternoon. My name is Tilmann Buerckstuemmer, and I'm the Chief Scientist of Myllia Biotechnology. And today I'll speak about a genome-scale CROP-seq screen, which reveals regulators of human T-cell signaling. Now, let me first introduce you to the CROP-seq workflow, which was initially developed in the laboratory of Christoph Bock, one of the co-founders of Myllia. This workflow is also known as perturb-seq or CRISP-seq or mosaic-seq. And so in this workflow, we combine CRISPR perturbation with single-cell RNA sequencing. And so to start with, we often utilize a cell line expressing Cas9. We transduce these cells with a guide RNA library. Importantly, this perturbation happens in a pooled format. We're strong believers in sort of a pooled format. And we then single these cells out using conventional single-cell RNA sequencing workflows, such as the 10x workflow that we're utilizing today. We reverse transcribe transcripts on the surface of little beads, and then we're able, sort of by pooling and sequencing, to infer which perturbation caused which transcriptomic phenotype. And I should say that this is sort of very heavy on the bioinformatics side and requires sort of dedicated teams and dedicated tools. Now, while this workflow is incredibly powerful, it suffers from the fact that it has sort of cost challenges, and consequently, most experiments that are published to date have been performed at rather limited scale. And so the ambition for the project that we're presenting today was really to get this to a genome scale screen. And so we thought of sort of two tricks that we could apply to get there. The first trick has to do with reducing cell numbers. Now, if you were to run a CROP-seq screen at a conventional, using the conventional Cas9 nuclease, well, then you would have to start processing about 20 million cells in one experiment. And now 20 million, even with a Chromium X, is really beyond the scale that's currently possible. And so we figured that maybe one way around this would be to put multiple perturbations into each cell. Now, if you were to do this with Cas9 nuclease, you would introduce 4 or 5, maybe up to 10 DNA double-strand breaks in a cell. And for most cells, that would mean that the cell would succumb to cell death. Now, this is different for CRISPR interference. CRISPR interference utilizes a dead Cas9 molecule fused to a CREP repressor. And if you target that molecule to the transcriptional start site of a gene, you can downregulate the message of that gene. And importantly, this does not need any DNA double-strand breaks. And consequently, it has been suggested and shown, in fact, that you can put multiple perturbations into one cell and still have a viable cell. And so with this first trick, by sort of putting multiple guides into one cell in a CRISPR interference format, we were hoping to be able to squeeze the genome-scale CRISPRi screen into just 1 million cells, sort of record just 1 million single-cell transcriptomes. The second trick was actually to apply targeted sequencing. Now, we're huge fans of the protocol that was published by Daniel Schreifvogel in 2020 in Nature Methods. And in this protocol, he developed a multiplex PCR, which allows you to pre-select the transcripts of your interest, amplify those and create a targeted library for next-gen sequencing. Now, this is particularly cool because it allows you to get away with less reads per cell in the experiment. So when we run targeted amplification as opposed to whole transcriptome amplification, we typically only utilize around 10,000 reads per cell, where a conventional whole transcriptome amplification setup would require around 50,000 reads per cell. So consequently, by applying this workflow, we were hoping to utilize much less reads for the entire experiment and thus reduce the cost for the experiment considerably. Now, to those of you who are not familiar with this approach, I just wanted to throw in one slide and show you how well this works in our hands. One thing I'd like to highlight before that is that we like the protocol because we can adapt it to our needs, meaning we can really customize the selection of mRNAs that we would like to amplify in the experiment. And so in order to assess how well this protocol works, this is a data set that's about 2 years old that some of you may have seen. We started from a cancer cell line for which we had bulk RNA sequencing data, and that cancer cell line expressed around 11,700 genes. And we put those genes into 7 different tiers, depending on their expression level, where tier 7 were sort of included the quantile 90 to 95. So those are the abundantly expressed genes. And it sort of goes down to tier 1, where we're really looking at the genes that are barely expressed in the cell. And so we took these cells and we subjected them to whole transcriptome sequencing on the one hand, and on the other hand to targeted sequencing, and compared the outcomes. And so what we're plotting here is the percentage of cells in which we saw at least one transcript, and we're plotting the whole transcriptome workflow on the y-axis against the targeted workflow on the x-axis. And what you'll immediately notice is that most of the dots, so each dot here is a human gene, and I should say that we amplified for each of the 7 tiers, we amplified 35 randomly chosen transcripts, and you'll immediately realize that most of the dots cluster under the diagonal. And so what that means is that the efficiency, the detection efficiency, is actually much greater in targeted sequencing than it is in whole transcriptome sequencing. It's sort of comparable for the tier 7, which is the very abundant genes, but it really is much greater for all the other tiers. And it really only levels off with the lowest tier, which is sort of barely expressed. It's in the quantile of 0 to 5%. And so we think it's an incredibly powerful tool. Again, it's customizable. So it means we can choose the genes we like, and we can design primer pairs really sort of at low cost. And this experiment suggests that in addition, it sort of is at least equally, often more sensitive than whole transcriptome amplification. Now, those were the 2 tricks we wanted to apply, sort of on the one hand, compress a lot of perturbations into one cell, and on the other hand, apply targeted sequencing to bring down the NGS cost for the experiment. That said, I'd like to introduce the model that we wanted to utilize for that screen and the signature we wanted to screen. The model we chose here is T-cell signaling, and that is because T-cell signaling is incredibly well understood. Now, we run a lot of screens in primary human T cells at Myllia, but for the scope of this experiment, which was really vast in terms of the number of perturbations we applied, we chose human Jurkat T cells. And in these cells, we can stimulate cells by applying a cocktail that stimulates, an antibody cocktail that stimulates the T cell receptor. And we first tried this cocktail on Jurkat cells, and we looked at an activation marker of T cells. This is CD69. And as you can appreciate, under steady state conditions, around 50% of the cells have that activation marker expressed on the surface as judged by flow cytometry. And when we throw in this cocktail of anti-CD3, anti-CD28, it leads to the upregulation of CD69 to around 94%. So this suggested that T cell signaling can be studied in these Jurkat T cells. Next, we wondered sort of how would we compose the mRNA signature? How would we compose the targeted primer panel that we were going to use for the experiment? And so in order to choose the genes that we wanted to amplify, we stimulated Jurkat T cells for 24 hours with this antibody cocktail, and we recorded changes in the transcriptome using bulk RNA sequencing. And as you can appreciate here, we're plotting sort of the total expression level on the x-axis against the log-fold change. Quite a number of genes changed in terms of their expression level. And we had roughly 1,600 genes upregulated and roughly 1,600 genes downregulated in the experiment. So it's a fairly profound change. And so from that, we then actually chose to compile the signature that we wanted to look at in the genome-scale CRISPR interference screen. And so we designed a panel of primer pairs that were supposed to amplify 374 target genes, and roughly half of those 374 markers that we wanted to amplify were dedicated to T-cell signaling. Some of them came out of the bulk RNA-seq experiment I just described. Some of them came from sort of a proprietary T-cell signature, and some of them were derived from looking at KEGG pathways focused on sort of T-cell signaling. But then what's really cool here is that we can sort of package additional screens into this one screen. And that is, we can choose other signatures that measure other biological processes, and we can query and question them at the same time. And here we made use of a paper that came out last year in Cell by Joseph Replogle and Jonathan Weissman's lab. And we looked at two signatures that were sort of in that paper and were quite prominent. One of them included the unfolded protein response, and the second one was focused on proteasome activity. This is the machinery that's needed to degrade proteins in a cell. And so we designed that set of 374 primer pairs. And again, this is an approach where we apply all of these in a pooled format. So it's not 374 individual PCRs, but really in a pooled format. And so then we introduced dCAS9 CRAB into the Jurkat cells. So this is the moiety that would actually lead to the downregulation of the genes targeted with the guide RNA. And so we brought that into cells, and we checked the activity by targeting a cell surface marker. Here we often like to target CD81 because we have a set of guide RNAs that work really well. And what you can appreciate is that on the right hand, we show a flow plot. If we're applying a non-targeting guide, 99% of the cells express CD81. And if we target a guide that sort of sends dCAS9 CRAB to the transcriptional start site of the gene CD81, then we get a roughly 90% downregulation of that marker gene. So it would suggest that CRISPRi is functional in these cells, and we can sort of proceed with a genome scale screen. So then we designed a genome-wide CRISPR interference guide RNA library, and that is based on Myllia's proprietary guide RNA design algorithm. The algorithm is not too dissimilar from published algorithms. It takes into account, on the one hand, the sequence requirements. It looks at the sequence we target, but also at the surrounding sequence. It takes into consideration epigenomic and epigenetic data, if and where available. And it has a strong component that is related to the position versus the transcriptional start site of a gene, and that has been described in the literature that there is sort of a favorable window where sort of targeting is most efficient. And so we target, with this library, we target 18,595 genes with at least 4 guide RNAs per gene. So that means we transduce with a library of roughly speaking 80,000 guide RNAs. And I had mentioned that our proprietary algorithm predicts, on the one hand, on target, but also off target activity. So obviously including guides that have multiple target sites in a cell. Okay, so this is sort of the preparatory work. Now let's look at the actual screen. So for the actual screen, we took the dCAS9 CRAB expressing cells. We took the guide RNA library that I had just described to you that targets 18,500 human genes. We transduced cells, and we then recorded the transcriptome using CRISPR, using single-cell RNA sequencing, and then on that data set performed the bioinformatics analysis. And so this is an experiment, as I had said in my introduction, that needs around 1 million cells at the very least. And this is where the Chromium X comes handy, because it's really the only instrument that's on the market today that can process such large numbers of cells. And so we processed 16 channels in that experiment, 16 channels of the Chromium X machine, and we retrieved around 930,000 cells from the 16 channels. And after filtering sort of for multiplets and for damaged cells, we ended up having close to 600,000 cells that were undamaged and that could be used for the experiment. Now, when we looked at the guide RNAs that were present in these cells, we found the distribution shown below here, which has a maximum of 13 guide RNAs per cell. So the median sort of in the experiment, the median cell had 13 guide RNAs per cell. Some had less, some had a bit more. And so as a consequence of that, the 600,000 cells that we recorded allowed us to have around 400 cells per knockdown. The other thing I'd like to highlight here is that we utilized a single NovaSeq S4 flow cell for that experiment. So again, it's a genome-scale CRISPR screen that requires just 16 channels on a Chromium X instrument and a single NovaSeq S4 flow cell to record the transcriptomic outcome. So the first thing we always check in these screens is whether we see downregulation of the genes we target. And in this case, we can only do so for the 374 marker genes that we amplify using the multiplex PCR I mentioned earlier. So here we're plotting the knockdown on the x-axis against the mRNA level on the y-axis. And what you can appreciate is this diagonal that shows up. And you know, if you look into this, roughly 94%, so 350 out of 374 marker genes, were successfully downregulated using dCAS9 CRAB. There was a small set of genes, around 24 out of the 374, for which we did not detect any downregulation. Some of those were 17 out of 24 were very lowly expressed, so we're not quite sure whether we're not seeing them sort of downregulate the genes or whether the gene is actually not downregulated. And then for a small subset, 7 out of 24, we possibly have a suboptimal guide RNA design and consequently do not see any downregulation of the target gene. But so the take home here is that for the great majority of the genes, we're able to downregulate gene expression quite significantly using CRISPR interference. So let's look at some of the phenotypes that we were interested in. Now, the first phenotype I wanted to focus your attention on is the unfolded protein response. Again, that was a phenotype where we benefited from Jonathan Weissman's paper. And so we nominated a bunch of genes here on the x-axis that in that paper were considered to be part of the, or sort of marker genes of the unfolded protein response. And what you can appreciate is that the knockdown of a number of those genes on the y-axis leads to the upregulation of these marker genes in the experiment. Now, the first thing we checked is subcellular localization. Now, for those of you who are not aficionados, the unfolded protein response really happens very close where translation is, and that is at the endoplasmatic reticulum. And lo and behold, most of the genes in that experiment were localized in the ER. It's also very evident that a number of those genes are sort of well-known suspects, including the genes SPC, S2, S3, that are part of the signal peptidase. So this is the enzymatic complex that cleaves off the signal peptide. There's DURL2, which is part of the sort of quality control machinery, as well as SEC61A1, which is part of the translocon. So really, sort of a lot of those genes seem to make sense. Next, we looked into a second phenotype. This was the proteasome phenotype. And here we saw something really interesting. And so first, what you can appreciate is that in all of those cases, we see a downregulation of the cognate gene, meaning if we perturb PSMD12, we see a downregulation of PSMD12. If we perturb PSMD14, we see a downregulation of PSMD14. Now, this sort of just tells us that the CRISPR interference machinery works. But then what we also saw in the PSMD14 knockdown cells is that a lot of the other proteasomal subunits were upregulated. And this is sort of a process that is really well understood and well described, and has been described, in fact, sort of more than 20 years ago, and is known to be a compensatory upregulation mechanism. And there is even the molecular mechanism of action is understood. There is a transcription factor called NRF1 that can sense sort of whether certain sort of imbalances exist, and that transcription factor then regulates at the transcriptional level the upregulation of other proteasomal subunits. So it's quite an interesting mechanism. And again, it was sort of revealed in this genome-scale CRISPR interference experiment. Now, what we were most interested in was T-cell activation. And so we took the cells in the experiment, and I should say that sort of the great majority of the cells, roughly 90%, were activated using this antibody cocktail that I mentioned previously. But there was a small fraction of cells that were untreated. And we first looked whether we could, on a UMAP representation of the experiment, whether we could distinguish activated from untreated cells. Now, the UMAP plot, what the UMAP plot does is every dot on that experiment is a cell, and it tries to separate cells by their transcriptomic phenotypes. And as you can appreciate, untreated cells cluster towards the left of this plot, where activated cells cluster towards the right. Now, next, we wanted to check if activated cells bear known markers of T-cell activation. And I mentioned CD69 in my introduction. This was the marker that we stained by flow. And as you can appreciate, CD69 is upregulated towards the right side of the plot where the activated cells cluster. So clearly, activated cells express high levels of CD69, which is sort of one key hallmark of T-cell activation. We then asked, so then we started looking into the perturbations, and we asked, can we find perturbations that shift cells from the activated towards the untreated population? And this is again sort of what the cells look like in terms of distribution. And there were two knockdowns, shown here as an example. There are actually more than two that very clearly shifted cells towards the untreated, sort of unactivated part of the graph, which would mean that the knockdown of those two genes really strongly diminishes T-cell activation. Now, in order to assess the T-cell activation phenotypes of the genome-scale perturbation more quantitatively, we established sort of a ruler, if you like, and we used an approach here that's called linear discriminant analysis that I'm sure our computational biologists could explain to you much better than sort of how it works than I do. But essentially what you do is you create, you create a ruler where on the left hand you have the untreated cells, and on the right hand you have the activated cells, and then you try to place each of the perturbations, each of the knockdowns onto that ruler in order to see if cells that harbor a given knockdown are significantly pushed either towards the untreated cells or towards the activated cells. And if we do this analysis, we find a number of genes that change the activation pattern of T cells. if you focus your attention first on the plot here on the left side, we're plotting the LDA score. This is the score that resulted from the assay I just described to you. And you appreciate that there is a number of, around 55 genes that are clearly involved in T cell activation. So if we knock those down, they diminish the degree of T cell activation. And conversely, there was a smaller set of around 15 repressors. So if we knock these down, then we actually see enhanced T cell activation. Now, a summary of those can be shown here on the right, lists sort of all the genes we found. And we try to highlight in bold those that are known from previous experiments and from the literature. And you'll appreciate that sort of a very significant number of genes in that set that was detected as significant has been described previously, which is sort of a great comfort for us. We did not run this screen in order to discover novel T cell biology. We really ran this screen as a proof of concept experiment, hoping to confirm a lot of the previously well-described regulators of T cell signaling. In fact, that was one of the reasons we chose T cell signaling that is so well described that we would sort of know by looking at the genes that we uncover, we will sort of know immediately whether the screen made sense or not. And the gene list presented here, we believe, makes a lot of sense. We can actually plot these genes onto the pathway. This is sort of the T cell activation pathway. And what you will appreciate is that many of them are really proximal, are either sort of part of the T cell receptor complex, it includes sort of several subunits of the complex, or really proximal to the membrane, sort of at the upper end of the signaling pathway. And again, the ones shown in green here are confirmed activators, and the ones shown in red are confirmed repressors whose knockdown leads to elevated T cell activation. The moment you sort of go down one level, we believe there is sort of a degree of redundancy so that it gets more difficult to find these factors in the experiment. And so again, we think it's quite striking, the genes that we're able to pull out in this one experiment as significant regulators of T cell signaling. With that, I'd like to summarize and conclude. We're very excited about this first genome-wide CRISPR screen that was sort of at our hand completed successfully. It recovers both known and new regulators of T cell signaling. What was important to us really is what we call here reasonable economics. We try to not perform this screen by throwing money out of the window, but really sort of on the one hand, we did the multiplexed CRISPRi perturbation so that multiple guides would end up in one cell. We did the targeted sequencing to keep the sequencing cost low, and we performed the screen on the Chromium X, and we processed just 16 channels. That was around 1 million cells, and that was sequenced on a single NovaSeq S4 flow cell. And why is this important? It's important because so far, a lot of people utilize the CROP-seq or PERP-seq workflow for target validation, but not really for target discovery because it was too expensive and it was really not sort of feasible to perform these screens at genome scale. With the experiment we're presenting today, we think that CROP-seq is ready to exit the niche of drug target validation and it's ready for unbiased drug target identification screens. With that, I'd like to acknowledge the people that have contributed to this project. The screen was run here at Myllia. Thanks a lot to Nicole Untermoser and Anke Loreger, who sort of performed the wet lab work. The analysis was done by Anatoly Vasiliev, Adam Krejci, with help from Lukas Badacher. And I also want to thank a lot our colleagues from 10x Genomics, Matthias Schranzhofer, Erika Urdaneta, Samantha Shelton, and Jens Dorothy for, on the one hand, generously sponsoring this, but also helping with a lot of discussions. So if you're interested in running your own CRISPR screen, please reach out to me. We're happy to discuss your CRISPR screening project and provide suggestions. With that, thanks a lot for your attention. I'd be happy to answer any questions you might have.
Excellent. Thank you so much to Tilmann and Angela for great presentations today. We're now ready to get started with our Q&A session. Please remember to submit your questions using the Q&A box on your screen. We'll do our best to get through as many of your questions as possible. All right, let's get started. Okay, excellent. There's a lot of questions here in the queue, so let's just get started. So we'll start first with Tilmann. Tilmann, for guide RNA ID, are you detecting the edits on target or are you detecting the guide RNA barcodes?
Yeah, for most of the guides, we're detecting the barcode. So we're detecting using the guide as a barcode and connect these barcodes to cellular transcriptomes. For a subset of the 374 for which we amplified the target mRNA, we were actually able to see if the guide RNA caused a downregulation. But that's really only for that subset of 374 genes.
Thank you. Moving on to the next question. This one's also for you, Tilmann. This person congratulates you on your work. And the question is, can you please expand on the convolution at the end of the pooled genome-wide screen? How do you associate phenotype to perturbation when you have a median of 13 guide RNAs per cell?
Yeah, so essentially the way this works is that we're grouping cells according to whether they have a given guide RNA targeting a given gene. And so it means if we want to compare the phenotype of the knockdown of gene A, we sort of take all the 400 cells that have that guide RNA targeting that gene. Of course, in that scenario that I just described, they will have a mix of other guides in there targeting other genes, but they for sure have those 400 cells all have that specific guide targeting that particular gene, or that set of guides, I should better say, set of 4 guides targeting that gene, and we're comparing those 400 cells to 400 cells that have a reference guide RNA. That could be a guide RNA target that is non-targeting or that targets a gene desert. And then we're comparing those two transcriptomically, and if there is a difference, we sort of call that difference significant and sort of ascribe it to the guide RNA that was in the perturbed set of 400 cells.
Thank you for that answer. Let's see here. Okay, also for Tilmann, the literature says that sgRNA design for CRISPR interference is more tricky and possibly dependent on the epigenetic state of the largest gene. This would imply that each cell line needs a different sgRNA library. Do you have any data to substantiate this?
Yeah, that's an important question because, of course, if you wanted to port this workflow into another cell type, and the guide design was very cell type specific, you would every time have to create a new guide RNA library, which if you're doing this at genome scale is sort of not fun. And so what we did in this particular experiment is we had a subset of genes for which we had good epigenetic information from Jurkat cells available. And we designed based on sort of that epigenetic information, a specific set of guides that was sort of a Jurkat specific sublibrary and screened these in parallel. And from comparing this Jurkat specific set of guides to the generic set of guides, we concluded that there was not a major difference between the guides that were designed based on the cell type specific features. Now, that doesn't say that there could not be very small differences, but we believe there are no major differences. And so we think that the library we created is likely applicable across many different cell lines.
Excellent. Yeah, I appreciate that response. Let's do one more question for Tilmann and give him a break, and we'll go to Angela for the following question. So for this one, I'm wondering why you chose CROP-seq over other techniques such as perturb-seq, etc. Is CROP-seq beneficial for transducing multiple sgRNAs per gene?
Yeah, that's a good question as well. So we chose CROP-seq. It's sort of in principle this workflow that I just described would be equally compatible with a non-CROP-seq architecture where we have guide RNA expressed from year 6 promoter, sort of without CROP-seq architecture. But then obviously, in order to detect guides, we would have to move to a 5' centered single cell RNA sequencing platform, which is well possible. The only thing is that in our hands, the primer panel design tool that was created on the back of the tool that Daniel Freivogel developed, that primer design tool for us it has been sort of more frequently used with 3' chemistry, and that prompted us to use 3' chemistry, which required CROP-seq. I think it's sort of possible to port this protocol and make it compatible with 5' chemistry, in which case the CROP-seq workflow would not be required.
Thank you, Tilmann. All right, let's move on to Angela for a couple of questions that we have in the queue here. For Angela, what's the difference between the single cell 5' and 3' CRISPR screening assay from 10x Genomics?
Yeah, great. So our 3' CRISPR screening assay, it basically requires that there's a specific capture sequence that needs to be inserted into the vector to actually capture the guide RNA. But for the 5' CRISPR assay, it's compatible with most pre-existing Cas9 libraries. So if you have an existing CRISPR library or you're starting single-cell CRISPR screening for the first time, we'd recommend that you could get started with that 5' assay, since it requires little optimization.
All right, and as a follow-up question, how does the sensitivity compare between 5' and 3' CRISPR assays?
So the sensitivity is comparable between those two assays, both in terms of the gene expression and the capture of the guides.
All right, thank you, Angela. Let's go back to Tilmann, because there's quite a few here left in the queue for you. All right, for Tilmann, you mentioned that you ended up with 400 cells per knockdown. Does it mean that 400 cells per guide RNA or 400 cells per target gene? Also, would you say that this number gives sufficient power to make assumptions on the perturbation effects?
Yeah, so we always design our guide RNA libraries to have 4 guide RNAs per gene. And so we ended up having 400 cells per gene, which means 100 cells per guide. The question of whether this is sufficient to sort of call phenotypes is not an easy one, because sort of in very general terms, sort of the number of cells in the experiment increases your ability to pick up phenotypes. Meaning if you had 1,000 cells per gene in such an experiment, you would be able to pick up very subtle phenotypes. And if you only had 50, you would probably only pick up very, very strong phenotypes. And so we felt 400 cells per gene in this combinatorial, if you like, screening paradigm was a good middle ground. But and it allowed us to recover a lot of mediators of T-cell signaling, which were previously known. So it would argue that it's sort of sufficient to pick up a lot of the known regulators of T-cell signaling. Could we go deeper by looking at more cells? I think the answer is yes.
Excellent. Thanks for that response, Tilmann. Let's see here. Let's keep going here with questions. Do you think the experiment would have worked if you delivered more guides per cell? For example, using a reduced number of cells?
Yeah, it's difficult to say where the upper limit of this approach is. I mean, honestly, when we started this experiment, we kind of aimed for around 5 to 7 guide RNAs per cell, but then ended up having around 13. And the way we gauge this is that we sort of look at the GFP positivity of the cells. We have GFP in the guide RNA expression vector, and we aim sort of for a very high percentage. But the truth is that the difference between sort of having 5 guides per cell and having 10 guides per cell is sort of a few percentages in GFP positivity. So it's not that easy to sort of always land exactly where you want to be. There is a very famous publication out there by Molly Gasparini that looked at sort of that connected enhances with genes. This is a paper that's 4 or 5 years old. I believe they even had more guide RNAs per cell, but they also had a slightly different cell line that was a K562 cell. So I think you can probably sort of put, maybe 15, maybe 20 guide RNAs per cell. I don't think you can easily put 50 or 100 per cell.
All right, thank you. This one is also for Tilmann. With so many perturbations per cell, how do you deconvolute? And was the median number of sgRNAs per cell optimal for deconvolution?
That's a good question. It's difficult to know what this would look like, if we had repeated the experiment with 3 guides per cell, 10 guides per cell, and 20 guides per cell, and see sort of where it starts being noisy. I think the truth is, again, if you wanted to uncover very subtle phenotypes, I think the preferred approach would be to have a single guide RNA per cell and end up with sort of very clean data. this is a compromise to sort of package more perturbations into less cells. And it clearly allowed us to recover known phenotypes, but I don't think we have enough data to say sort of what is there sort of an optimal number of guide RNAs per cell that that is still sort of reasonably sensitive, if that is sort of what the question is.
All right. Thanks, Tilmann. Yeah, if the person that asked the question have a follow-up, feel free to put it in the Q&A box, or you can email us afterwards and we'll try to get back to you directly as well. All right, so one more question for Tilmann, and we'll go back to Angela. Tilmann, how do you make sure that the perturbations that are observed when multiple guide RNAs are present in one cell are not combinatorial, and that perturbations of the guide RNA separately would have different effects?
Yeah, I think they could potentially be combinatorial, but then because we have a very large guide RNA library within 400 cells, we would never find the same two genes perturbed, or very few cells for the same two genes perturbed, meaning we would end up with so few of those cells that if any combination of two had a very strong phenotype, we would probably miss it. So the only way to sort of analyze this in a reasonable fashion is to sort of look for all the 400 cells that have a guide targeting the same gene and assume that the phenotype that is common to those 400 cells is the phenotype that's caused by that perturbation.
Okay, thanks for that response, Tilmann. Let's go to Angela now. For question for Angela, how does single-cell CRISPR screens differ from bulk CRISPR screens?
Yeah, so when you do single-cell CRISPR screens with 10x technology, it basically will capture and barcode the guide RNA and also the transcripts within each cell. So it gives you a direct link essentially between the perturbation and then like the phenotype. So the readout gives you that information for each single cell. And so what that does is really gives you that resolution to really cluster your cells and identify different subpopulations of cells within your sample.
Great, thank you, Angela. Now we're back to Tilmann. Tilmann, how did you determine the optimal time point for single-cell processing after library introduction?
Yeah, that's a good question, and we often debate this, because obviously you need to give the perturbation a bit of time to sort of kick in, and particularly with CRISPR interference, but also with other CRISPR perturbations, you're sort of fighting a little bit against the protein half-lives, against the half-lives of mRNAs in the experiment. So a good guideline in let's say, at least in proliferating cells that we worked with in the past, is that the earliest time point we're usually looking into sort of CRISPR phenotypes is probably at day 7. And then sort of for CRISPR interference, we usually like to wait a few days longer. So typically until day 10. And then obviously the other thing that we made sure is in that set of pre-experiments, we determined a good time point for sort of post T-cell activation. And sort of obviously that was determined by bulk RNA sequencing to be sure that the transcriptomic signature with screening is still sort of up or down-regulated to a good degree and can be captured with single-cell RNA sequencing.
Great, thank you, Tilmann. And it looks like we're down to our last couple of questions here, and they're both for Tilmann. Tilmann, what are the advantages or disadvantages of CRISPR interference over CRISPR knockout screens?
Yeah, generally we like, for loss of function screens, we like CRISPR knockout screens better because they give very clear phenotypes, right? The moment you knock out a gene, or at least sort of ideally, if you knock out a gene, the phenotype is crystal clear and it's sort of absent in a gene knockout, it's present in a wild type cell. CRISPR interference gives sort of more in between phenotypes. Conversely, with CRISPR interference, you have a chance of capturing phenotypes of essential genes, which in the CRISPR knockout screening paradigm would sort of otherwise disappear from the experiment. The other sort of added benefit, and I've highlighted this in the talk for CRISPRi, is that it allows multiple perturbations per gene. And that was really the starting point or the starting trick as I indicated, for being able to squeeze a lot of perturbations into one cell. But clearly, if I had unlimited resources, right, I would prefer a CRISPR knockout screen with a single guide per cell. But that would then mean that instead of sequencing 1 million cells, we would have to sequence 20 million cells, which is sort of an expensive endeavor.
Yeah, I appreciate that insight. And we are now down to our last question. This one's also for Tilmann. So Tilmann, which features are taken into consideration for designing the primer panel, and how much does a primer panel typically cost?
Yeah, I'll ask my colleague Anatoly to help me with that question. I can tell you about the cost. The cost is sort of very low. It's sort of a nested PCR protocol, and sort of you design 2 primers, the conventional primers, and the cost per primer is in the order of magnitude of €5. Anatoly, you want to speak quickly to sort of some of the key features? I'll put the camera over to you.
Sure. Hello. So the features that I like to mention are, in the first place, the poly-A site coordinates, because the primers, they need to be designed in the close proximity to the poly-A sites. And unlike other annotations, this data is not readily available. So we calculate the poly-A sites from single cell or bulk RNA-seq data, which is 3' biased. And the other features that might be taken into consideration are the specific gene isoforms that are expressed in the cell type of interest, because the primers are designed in the exonic regions of the genes. And probably the last important feature to mention is the primer's compatibility in the multiplex PCR setting. And there is a computational tool that allows you to do that.
Brilliant. Thank you, Anatoly.
Excellent. Well, thank you, Anatoly. Thank you, Tilmann, and thank you, Angela. That wraps up our Q&A session for today, and it wraps up our webinar. I'd like to thank the audience again for joining us today and for your questions and engagement. If we didn't get to your question today, or if you have any follow-up questions for us, you can send us an email at the address support@10xgenomics.com. In a few moments, you'll also see a survey pop up on your screen. We'd really appreciate any feedback you can provide for us, and we can learn a little bit more about your research goals. Thank you everyone again, and I hope everyone has a nice rest of the day.